Hi, everyone. Thanks for joining the third in our series, the Go with the Flow Expert webinar series. I'm just going to wait a couple minutes here to allow some more attendees to join us, and then we'll get started. So thanks for joining. Okay, hello everyone. Thanks for joining the third in our Go with the Flow Expert webinar series. Today we have Jay West, Principal Scientist in Biologics Process Development at Bristol Myers Squibb, here to talk about the FlowVPE and FlowVPX, our new inline system for measuring concentration. The first two of the webinar series are available on demand now. You can just use the same registration link to go back to the page, watch the first one on DVC measurement and the second one on impurity measurement with the Malls detector. Today we have Jay, which we'll get started in just a minute, but just as a reminder, next week at 8 a.m. Pacific, we will have Raf Dedier from Janssen to talk about the drug product work that he's done with the FlowVPE. And then finally, the last in our series will be a panel discussion with Tony Hunt, our CEO, about these various presentations and the system. So without further ado, here's Jay West to talk about the FlowVPX, FlowVPE for UFDF. Thanks, Jay.
Thank you, Ramsey. Sorry, I just wanted a couple of logistic things. Make sure, first, can everybody hear me?
Yes, we can hear you.
Okay, great. So the volume is correct. And so can you see my slide number 1, evaluating the new CTech and webcam?
Yep.
Okay, good. So let me just go back. So everything's still working? And then I'm just going to go forward one slide and make sure that works. Does that work?
Yes, we're good to go.
Okay, perfect. Thank you for verifying that. Okay, so welcome, and thank you, Ramsey and Repligen, for inviting me to give this expert webinar series talk. And the title of the talk is Evaluating the New CTech FlowVPX System for the UFDF, monitoring protein concentration. There's a second title for the talk. So that was when I was invited to give the talk. I think it was sort of Repligen and I went back and forth, but I actually want to broaden the scope of the talk. So the focus of this talk was a 6-month beta test we did of the new FlowVPX system, which, as people hopefully know by now, is meant to replace the FlowVPE instrument, which is about a decade old. And we started actually and did this beta test evaluation before it was commercially released, starting last year. So that's the focus of the talk, but I want to talk a little bit more about PAT in general, because that's going to give you a better scope of why this is an important instrument in enhanced PAT technology and how it could change the bioprocessing significantly once it's implemented, and start the process of enhanced PAT sort of taking over as a new paradigm for process analytical technology. So the broader title is using the FlowVPX for inline UFDF protein concentration monitoring. So a little bit about me. I've been at Bristol Myers Squibb for 7 years now. I started working on PAT about 5 years ago in the process development analytics section of within the process development here at Devens, Massachusetts. So this is a new campus that was built about 10 years ago for Bristol Myers Squibb to do biologics development on the commercial, basically to develop the commercial manufacturing processes. And I became more heavily invested in doing PhD research about 4 years ago. I joined the BioForum. Everybody knows that's a consortium. So now at the point where I spend most of my time doing process analytical technology development at Bristol Myers for the biologics department. Okay, so first of all, the FlowVPE/X. This talk is about both instruments and it's part of the story. Like I said, because we actually started using the FlowVPE in 2017, and I want to talk a little bit more about the history of that. But I want to show you how it sort of fits into the overall scheme of biologics manufacturing, particularly at Bristol Myers Squibb, and how that's been evolving over the past decade, especially since I started 7 years ago. So first of all, I want to start from the upstream portion and how that's changed. We're not several years ago, the titers were about 3 grams per liter. Now we're up to about 10 grams per liter. So the output has significantly increased. The next step in the process that's made a significant change is the protein A, which is multi-column chromatography capture. We use multiple columns to make the protein A more efficient. The next step is automated viral inactivation. That used to be a manual process that has becoming more and more automated. And as you notice, there's a theme here of increased automation throughout the process. And that's where the FlowVPX will come in at the end. So the polishing steps, that is also as part of a general overall trend, the biologics is moving towards more continuous manufacturing. And at BMS, we've incorporated elements of continuous manufacturing. So the polishing operations is something that's undergone some continuous manufacturing research. Now to get to the UFDF and the FlowVPX and where that fits in. So as of present, in say our biologics manufacturing, and throughout the industry in general, there is no inline monitoring of the protein concentration during the UFDF process, save for maybe some early adopters of FlowVPE or VPX I'm not aware of, but at the very least, from my knowledge of the bioform, nobody is controlling the UFDF process based on protein concentration. And that's significant because the UFDF operation has been undergone significant changes itself in the last decade. What happened was there was an evolution from intravenous injectables in biologics to subcutaneous. And as that's happened, the concentrations of the drug substance have increased significantly to where your operating concentrations are up over 200 grams per liter, which is several times higher than previously. Previously, drug substance was formulated at about either from 10 to 50 grams per liter. Now we're going up to the 150 to the 180. So at present, the control of the UFDF is done by measuring mass. You take an initial concentration of the starting material, and based on the change in the mass of the system, you extrapolate what the protein concentration is. So is anybody who's familiar with protein at very high concentration and their biophysical properties, you're very close to the limits of solubility in the 200 to 250 gram per liter range working with a protein. So it stands, basically, to me, it's almost self-evident that if you can control the process based on protein concentration, that's a very significant advance in the potential yields of your process, consistency, and especially, like I said, you're near the very limits of protein solubility during your process. So, which gives a very strong reason why an inline A2 instrument would have a significant amount of value. And so let me so focusing now on the VPX and the UFDF process, let me show the bottom what happens during the UFDF process, just for those who are not familiar. So after viral filtration, the material is about 10 grams per liter. Then it's concentrated. This is the modern process for biologics for subcutaneous platform. You concentrate to about 70 or 80 grams per liter, diafilter with about 6 turnover volumes of formulation buffer, and then concentrate to a target called the UF2. What that is targeted to be about 10–20% above the target concentration for formulated drug substance. Because then what they do is they drain the system, as you can see in the diagram, like, sort of like a bag. is either by pressure or gravity. After it's concentrated, the UF2, it's collected in a bag. Then you push buffer through, which represents the dotted line as the concentrate. So that dotted line at the very end after UF2 represents the hypothetical protein concentration, basically, as you recover the material and push the buffer. And it's basically diluted into the purified drug substance concentration. So the key is, if you can control that UF2 concentration with an instrument like the FlowVPX, you can basically collect your material consistently at a very precise purified drug substance value, which is the key element in the value proposition to the VPX. And I'm going to talk about that in the next slide. So why why the VPX? Why is that such an interesting and potentially interesting instrument for GMP use for inline? So first of all, the FlowVPX has a very large dynamic range, which I'll show the data for. It can accurately measure proteins from 1 to 300 grams per liter, which I'm not sure there's any other technologies that can do that other than say the FTIR or Raman. Those instruments, you have to build a model in order to calculate protein concentration. So an advantage of the VPX is it's kind of basically plug and play. It really does not take long to obtain accurate protein concentration values once you add your instrument to the UFDF. The other one is its proven technology. So as I've shown, as the process evolved and it's become more modern and advanced, the one thing that's lagged behind is process analytical technology. There's really very few, is what we call enhanced process analytical technology tools that are used in the GMP space right now. And so this VPX actually would represent one of the earliest forms of what they call enhanced process analytical technology for basically control of the process. That's why it's a quite exciting field to be working on now, especially considering the potential. Next slide. Okay, the potential benefits of I say the FlowVPX. This would be for, I just want to, as a disclaimer, this is not an advertisement for the technology. This is, but I'm going to show data that shows that this instrument has a lot of potential, and that basically you could sort of back that out and just say any inline measurement of protein concentration, how that would benefit drug substance manufacturing. So first of all, it's going to decrease the process variability and product loss during UFDF and recovery. It'll increase the process data and consistency, and it's going to eliminate offline measurements. So if I didn't mention it already, right now, in most manufacturing spaces for biologics, or at least at Bristol Myers in our clinical suite, what happens is when you take a sample of the recovered material that's been diluted, it goes to the QC laboratory for an offline measurement that takes a considerable amount of time to get that result back, so typically several hours. So there's a movement to use the solo VPE, which can basically decrease the amount of time waiting, because you need that value to forward process, because you cannot formulate your drug substance until you know that the concentration is in the target range, because once you've added your excipients, such as like PS80, those can't be, you can no longer reconcentrate your material. So you have to absolutely be sure you're in the correct range before formulation. That's why these measurements are so critical, because there's no turning back from once you've made formulated drug substance. So you're going to potentially eliminate the need for offline measurements. Though, like I said, we're going sort of in a stepwise strategy in terms of GMP, that there's going to probably be a transition period where it's going to be the FlowVPX and the solo at-line measurements that would be the next step in the evolution, and then followed by ultimately possibly use of a FlowVPX or other real-time A280 measurement system in order to basically completely automate that whole UFDF process all the way through making formulated drug substance. And number 5, it's critical, the alignment of technology throughout the entire process and across manufacturing sites. So as Repligen is scaling up production of these flow sensors, it's a potential that you can put these from site to site, and assuming they can consistently build these instruments to have very high precision from instrument to instrument, that can help quality assurance at different manufacturing sites for when you transfer this process to different places on the planet. So we, as I mentioned before, we started working on using the FlowVPE in our UF/DUF development all the way back in 2017, that first generation instrument. So along with the improvements in the technology, and it's like the technical parts I'll explain later, there's actually we've had discussions with Repligen that there's a lot of interest in putting this into the GMP space. And anybody who works in the GMP space understands that these systems have to be enclosed and be water resistant. So as you can see from the 2017 picture, the head unit of the VP is sort of sitting on top of the UF/DF skid. There's a carry 60 spectrophotometer on a cart, and then the laptop is open. And that's basically, the laptop is controlling the instrument. The carry 60 is the detector and providing the light source, which is critical for the VPE. Then you see 2019, CTech built a cart to enclose the carry 60. The Tough Book is closed. There's actually a new generation Tough Book, and there's potential for putting this, eliminating the Tough Book, or at least it can be put into that cabinet and away from the UF/DF skid by operating through SyntiQ. So basically, as it's evolved, the FlowVPE makes the first step towards GMP. The VPX has even further steps in the change of the hardware and control systems to be all enclosed in a box that hopefully is waterproof and automated into Delta V. Okay, so this is sort of you can find this probably on the Repligen website. And I'm not going to go through every single checkbox comparing the VPE to the VPX. Suffice it to say that I'll jump to the conclusions that the VPX is a significantly superior system for GMP use and a step forward. And I'll just sort of go through the list and verify the points that I know of to be true for the VPX from the VPE and the VPX. So in terms of the 5-second cycle acquisition time, realistically, you can obtain spectrum, or I'm sorry, concentration about every 10 seconds, which is pretty much fast enough. You could probably get down to 5 seconds, but we typically observe about a 10-second cadence between readings, which is plenty fast for UF/DF. And in fact, the strategy, if you're measuring inline and you want to do, say, make a decision, maybe probably the best strategy will be to average several of those points before forward processing. But that's another part of the strategy. I don't want to get into details right now. So there's been a change in the software to something called Viper. That has just been released along with the VPX head unit upgrade. And it replaces actually just like an earlier software that was built on the Carry 60 shell. And the Viper software is going to be CFR compliant. That's not something we've spent much time developing yet, so I can't report back on how much of an improvement is. But they promise that there's going to have OPC output. The digital input output, that's been something that you can do with the VPE. We've done that before. For simpler small scale UF/DF setups, that's something that may be a better choice if you're setting up small scale UF/DF. But OPC will be with the large scale skids. So let's go down to the concentration range. That's really the most critical thing in the development of the VP to the VPX that allows us to seriously consider this as a GMP instrument. Back when we were running the VPE, one of our particular assets in our portfolio, we could not measure accurately above 150 grams per liter, which sort of killed the idea of using the VPE for UF/DF and GMP, because it's such an important asset that there was no reason to spend a lot of money in implementing VPE into our commercial and clinical UF/DF if it wasn't going to work for some of our portfolio assets. The VPX, it looks like we'll be able to use potentially all our portfolio assets for UF/DF, because the concentration range has gone up to at least 260 grams per liter. That's what we verified. So that's the biggest, probably single technical improvement. The flow cell size compatibility, I guess they're going to increase the number of options there. So the critical step forward for the VPX was that they decreased the path length step size from 5 microns to 1 micron. And I'll show you, okay, that it's in a couple more slides, that technical chart. So starting I believe it was July of last year that we first obtained the VPX system, and we had VPE. So we decided to test them in series, head-to-head, first of all. So this is just some quick pictures of the beta system. Actually, it looks slightly more refined. We obtained the first VPX commercial instruments recently, and they look a little prettier, but essentially it's this cube on a stand. So that is, from a GMP perspective, superior, because the VPE system, as you can see, has more like exposed doors and parts that could be tripped up in a GMP manufacturing environment. So the VPX, it's kind of basically a sealed brick on purpose. So this is what they look like side by side. This was an early small scale UFDF experiment we did, and I'll show the results of that. It was to evaluate the VPE versus the VPX for, I'll call it monoclonal antibody 1. That's like I said, an important asset in our portfolio, and it was critical that whatever A280 system we use for measuring UFDF, be able to measure that asset accurately. And the reason for that too is that particular antibody has a very high extinction coefficient. So that's on this next slide. Okay. So here's sort of the issue we were running into with the previous VPE, and I'll call it monoclonal antibody 1. So this is actually solo data I'm showing, but it basically it gets to the point of the limitations of the previous generation VPE. So the solo VPE and FlowVPE both have 5 micron minimum path steps. However, the solo VPE 0 point, the lowest, the shortest path length is 5 microns. When the FlowVPE, you cannot get a 0 of 5 microns just due to the way that the instrument is designed. The 0 is actually more like 15 microns. So what was happening, so say at 200 grams per liter, the extinction coefficient is around 1.7 for this. So what happens is, you can see when you do, this is the solo VPE, starting at 5 microns, when you get above 30 microns, Beer's law is no longer linear. So when we would run the FlowVPE, if 15 microns was your starting point and you use 6 points, we basically were getting inaccurate protein concentration readings, which the FlowVPE would never be able to accurately determine for this monoclonal antibody due to its high extinction coefficient. However, as you can see, the solo VPE, because it has the 5 on the far right, when you cut it down to 6 points that you fit the curve to for the VPE, you can get an accurate concentration reading. The problem is, like as I said, that the point number 1 is 15 microns instead of 5 microns. So that basically, it demonstrates that there's a limitation with the FlowVPE. It's actually significantly more limited than the solo VPE. So what they did at Repligen is they cut that down to 1 micron. And I don't have the curves, but we witnessed basically that the path lengths that it chose, for example, for this protein when it's measuring the concentration above 200 grams per liter, it typically would select 8 to 13 microns as the range to determine concentration, which is perfect because that you're in the linear region of Beer's law and gets around that issue. Now here's some data where that first small scale experiment to show you what the data looks like when you go over 150 grams per liter with the next gen, the VPX, that's at the bottom, versus the previous generation FlowVPE. And it had the R squared on the right. So first of all, the VPX readings were in cycle counts. Because of an early technical issue with the Viper software, we couldn't output the data in time. But essentially, this actually shows that 1,000 cycle counts is approximately 200 minutes. So you're doing 5 cycles per minute, with about every 12 seconds you get a concentration reading. There was some bumps in the beginning during the concentration. There was actually some air in the system. But as you can see, the VPE and VPX, very similar all through the diafiltration chain phase. Once you get up over 150 grams per liter, the VPE actually started kind of fluctuating wildly. And in fact, it gave false readings because in the algorithm, it actually has a search algorithm to try to figure out what path links to run each time. And it does a sampling first and then takes the actual data. Because when you get over saturation, the algorithm does not function right. So we can't pick the right wavelengths, path links, excuse me. So there's no other word to say it. It kind of goes haywire. And as you can see, the R squared starts to, once you drop below 0.999, in my experience, you no longer can obtain an accurate reading for a monoclonal antibody. However, the VPX in this first section, it went all the way up to almost 250 grams per liter, and the R squared stayed above 0.999. So here's the next slide. So this was a sort of a verification of the VPX. So what we did in this next experiment, we took the VPA out of the loop and just did a straight VPX. And we took several samples during the study to test offline against the solo VPE and the A280. So I want to just mention, because this is again is the MAB1. So the solo VPE can measure this MAB1 up to 240 grams per liter accurately, but that's kind of the limit. And again, because of that high extinction coefficient, it runs out of room above 240. That's from qualification data from another department. However, the A280, so that is a gravimetric assay that's used by quality control. And what that entails, that's the one that takes several hours to complete because you're essentially diluting approximately 50 micrograms of material into 20 mils of buffer. So it's basically a pain to sort of do, and it takes, you need to know the density data. It's not the easiest technically assay to run. So that's another reason why the FlowVPX would be a significant improvement, because it would eliminate this need to do this complicated A280 dilution process to get an accurate protein concentration range. And based on the data, there was good agreement between the three methods, like I said, all the way up to 240. And at that point, the FlowVPX gave a reading of 260. The so-called actual, which is like the gold standard, was 257. So all throughout the concentration range, the VPX actually biased slightly high. And we're not entirely sure why that is. In a crude way to put it, it could be because the readings are of fresh material. Because once you, one of the issues we've run into too is this material before it's formulated can be slightly unstable and start to precipitate. So that's one of my at least hypotheses why we kind of saw a slight bias towards the high end with the VPX versus the offline methods, is that when you remove a sample, you can have precipitate form sometimes, or just it's sometimes difficult to remove a sample that's completely homogeneous. In any case, except for the reading at was it 72 grams per liter, it was within 5% of the other offline methods. And I said the solo VPE reading, I said, would not be considered accurate because of the limitations of that instrument for this molecule. So I just want to say, for a GMP instrument, the benchmark, it has to be absolutely within precision and accuracy 5%. 5% of basically plus minus 3 standard deviations for the precision would be considered acceptable. Typically, the standard deviation is about 1% for these VPE instruments. So so basically you're looking at plus minus 3% variation. So check mark in that box. It's within 5%. The other one is accuracy, and it appears that it is within 3%, because 5% is the absolute limit. But if you're talking absolute limits, you actually want to be closer to 2 to 3, which is based on this data, it appears that it can achieve those benchmarks. And I have a couple more slides showing the data from the studies. So this is for another monoclonal antibody that does not have such a high extinction coefficient. And in fact, I'll mention too that the FlowVPE was able to previously measure this molecule accurately as compared to the offline methods up to 250 grams per liter. But we wanted to verify that the VPX was also accurate for this molecule too. And as shown, we did solo VPE and FlowVPX tests. There was not a full UFDF. We actually just did a small scale concentration and removed 5 samples for offline testing to compare with the FlowVPX. So I just want to add to the FlowVPX readings. The way we took the readings is we would stop the system and let it recirculate until it achieved an equilibrium, which basically meant a couple minutes where the VPX gave consistent readings. At that point, we would mark it down in the time log, and I would average 1 minute of data, and at the same time, we would pull a sample from the circulating material that was essentially at equilibrium, and then as quickly as possible do those offline measurements. Because, as again, I said that this material was unstable, so we did it always within like 12 hours of pulling that sample, we would do the offline measurement. So here's the result of the beta test. So we did 10 full UFDF runs with 6 biologics. So not only monoclonal antibodies, but a couple of fusion proteins. We tested in series with the FlowVPE. The conclusion was, at least for this monoclonal antibody one with a high extinction coefficient, the dynamic range maximum was 150 grams per liter. So that's a fundamental limitation that we cannot see any technical way to overcome, except with the exception that we considered you could change the wavelength that you measure. For instead of using 280 nanometers, we actually look at the possibility of using 250 nanometers. In that case, the FlowVPE could read up accurately up to 250 grams per liter. However, almost like in order to best align with the offline gold standard, we really wanted to stick to using 280 nanometers as the wavelength where you measure protein concentration, because that's well established. And 250, as far as I know, is not used to measure protein concentration. But that is a workaround for the VPE, potentially, if people want use for the older instruments. So therefore, the VPX, we were actually able to measure up to 260 grams per liter for monoclonal antibodies. And so I just want to show for the data from the table. We did not do A280 on each one of them because those experiments are not sometimes time-consuming to run. But we measured at least solo VPE because we know the solo VPE can give accurate readings, as again I said, up to 240 grams per liter. Sometimes it will read slightly low for A280. However, so monoclonal antibody 1, this was the 3rd UFDF. The solo VPE, again, trended slightly lower, but was in 5% of the VPX readings. Monoclonal antibody 2, you already saw that data. We were within 5% of the solo VPE and even closer to the A280. Monoclonal antibody 3, we were within about 2% of the solo. Monoclonal antibody 4, that was within 3%. And then these 2 fusion proteins, we were within a few percent of the solo VP. So in nice agreement. So so far so good. The data looks good. This was only one instrument, and we've actually ordered some new instruments. We're going to be testing them soon to hopefully ensure that you get comparable results and precision from instrument to instrument and flow cell to flow cell. So the development team, I'm probably going to be missing some names. This spans back 4 years, and it doesn't include like all the people that have done work on it. My apologies, but I really just want to take it on PDA. Sue works for me. She's been helping the last 4 years. Letha is my supervisor. She's been helping me with the project. Danuka joined the group a couple of years ago as a PAT SME. He's especially going to help me with getting the GMP implementation. Julia is the director of the group, and she's in basically the executive support that I need for the project, and has been very supportive all along. Neha Puri left about 6 months ago. She did a lot of the work for the beta testing, the data I showed. So just going to downstream quickly. Melissa is the head of the UFDF here in Devens. Haseen and John have helped with experiments. The scale-up lab downstream, I can't thank them enough. They've been essential So the last, they actually adopted the FlowVPE for use before I was even involved. That was largely due to Derek and someone named Nirpan Singh, who's moved on to an MST department at Bristol Myers Squibb. DigiJ is helping with the project now. Danielle has in the past. MS&T has been involved much more recently. Just quickly, so G is really leading the PAT efforts and along with Spencer, and he reports to Dong. Kaylin did some work with the VPE previously. She actually worked for me and switched departments. Manufacturing Technologies is also interested in this for the commercial use. Tanushri has been involved along with her co-op Catherine. Within so the single-use facility is sort of our GMP clinical suite, and Nancy and Nai have been helped so far along with a number of other people. There's also efforts going on in New Brunswick, New Jersey to use this for drug product GMP manufacturing. And like I said, I won't go through all the names, but Peter has been leading that effort. Has been doing a great job on that. And we're sort of supporting each other, like I like to think of in terms of buttressing each other in this project. And it just gives you an appreciation too that even though this is a simple, I'll say relatively simple technology, that getting this into GMP is a relatively daunting task. But Repligen so far has done a good job in upgrading the VPX so that we can realistically consider putting this into a GMP facility within the next few years, best case. I haven't talked about the flow cells, but again, with Repligen, I want to thank them for cooperating with us over the years and listening to our feedback on how to improve their instrument. I think a lot of, at least according to what they tell us, that the feedback we gave them from the FlowVPE was used to create the VPX instrument. And so far, like, we're pleased with the results and look forward to working with them in the future. So yeah, just one more shout out to Repligen. Like I said, they've been very good to work with. They have listened so far to our feedback and are trying to basically move this forward as quickly as possible, hopefully, so that they can, have this instrument be one of the class leaders for enhanced PAT instrumentation, which, like I said, there's not really a lot of instrumentation available at this right now for enhanced PAT. So it's been an interesting journey so far, and we're still a few years away from GMP. I think, or hopefully maybe sooner rather than later. And you're going to hear about, I think, some other efforts in the next few weeks about GMP implementation. Thank you very much. Yeah. So any questions? I think Ramsey's got some questions. And,
yeah. Thanks a lot, Jay. Thanks for the informative presentation and the story at BMS. And I mean, thanks again for all the support from everyone on that list. It's been really fun to work with you guys. So as a vendor, it's been great to be welcomed with open arms at a customer and really get to do some real work with some real future implications. So I'll jump right into the questions. So we already have a couple. So if people have questions, please put them into the questions box and I'll read them out and Jay can respond. The first is, do you have plans to implement it into production to marked or only in small scale GMP? What are the timelines? I'm not sure if she meant that to say production to marked, but maybe she's asking large scale or small scale GMP. Autocorrect is kind of a pain in the neck.
Sure.
So what do you think there, Jay?
Yeah, I can, and like I said, hopefully, if I'm being a little cautious, understand that the nature of what we do is somewhat proprietary, and I believe, hopefully, in sharing as much as possible, because I forgot to, sorry to back up. I also want to thank the Bioforum too. I've been part of that for 4 years, so that's a lot of actually of the spirit of like sharing this knowledge and wanting and to have an industry sharing. I think it's a great thing that we're doing. That I know that when you work in the biopharmaceutical industry or pharmaceutical industry, there's this balance between what's proprietary and then what we can publicly share. And I always try to adhere to the spirit of trying to share as much as possible, because it's a reciprocal thing. That when someone rises, we all rise together, right? It's good for everybody, especially things that, and the reason that I'm willing to share, I think, so much of the development of this instrument is that we're hoping, just if this isn't self-evident, that the more people that adopt this, the easier it is for the regulatory authorities to sort of give their blessing to this. So we're sort of all in this together. That's part of the mission of the Bioforum, which I've been working with for 4 years. It's a consortium of biopharmaceutical companies. So with that preamble, I'll answer the question. Basically, so that we, at present, we're trying to work on clinical and commercial manufacturing implementation, but I only go so far as for process monitoring, because right now there's really a gap in the amount of information we have in the process from an analytical perspective. So before we can control the process, which like learn to run, basically we need to learn to walk, and which means that we need to get this in there and do process monitoring first. So right now, as we learn to walk, we're trying to get this into the clinical manufacturing and commercial manufacturing and sort of demonstrate its bona fides, and then go ahead with more ambitious goals such as control the process and automation. But I think that, to be honest, yeah, it's kind of a baby step thing.
So there's a follow-up question to that, Jay.
Yeah,
what are your main concerns for the GMP implementation?
My main concerns is just getting buy-in from all the stakeholders, because this touches so many different stakeholders in a pharmaceutical company that you can't. It's going to require very high level sign-off, executive level sign-off, in order to implement it. It's not just something simple that you can sneak under the radar and throw in there. It's this touches like the highest level of quality, because I said that it's, if something, whenever you're, as anybody who's worked in the pharmaceutical industry for long enough knows, it's an inherently conservative industry, because the consequences of instrumentation, say causing an issue with quality, are enormous, because our products are injected into human beings. It's not like a plastic. So that's one of the things too to appreciate. It's like you look at the chemical industry at large, we're behind in process analytical technology, but it's due to caution. Basically, is that if you're manufacturing a piece of plastic and the quality is off for a lot, it's not a big deal, whatever, they recall it. But you can't, it's almost incalculable the damage to a pharmaceutical company's reputation if you release a batch of say drug that's not what it claims. Like so, and this is going to be part of the batch records going into with the drug substance. So it absolutely has to be accurate. You can't have, if reporting on something that's going into the FDA filing, that you have a process, or you're using analytical instrumentation that's where it's not 100% almost sure that the measurement is accurate. So, and one of the actually, so what is it? Actually, here's a technical thing that hurdle that we're trying to overcome. So you have, say, for example, we've sort of aligned on the use of a single-use flow cell because it's so far down the process. So we're trying to avoid having to do cleaning, validation, and reuse. Anybody knows too that as you get further along the process, it gets cleaner and cleaner. You want to avoid bioburden because you're getting very, very close to the end. And the way you typically do that now, especially with these, when you have a clinical suite, you're going through a lot of different molecules, you're switching over, is to use single use. So in the case of single use, you need to, you're hooking up a single use flow cell to the instrument. And how do you test something that it has to be absolutely clean? So typically, with an analytical instrument, you'll basically add, some material to it, or like, an HPLC, you'll inject something, it shows that the instrument's working properly. How do you do that with an absolutely dry cell? Like, how do you know that is like going to measure your process material accurately? There's ways to do that. It's just something that we need to work on to ensure that we can take a brand new untested cell hook it up to the process, and that it's going to be within 2 or 3% of actual. So that requires like some strategizing with QA, QC, some clever, analytics that haven't really been considered before, because that's part of the system suitability. When they do system suitability for offline systems, it's a very elaborate procedure. So to do it on an inline system where you're swapping flow cells, it's kind of a new paradigm that we've entered. And I think, yeah, I know the next talk, Rolf might be talking more about that, how they sort of figured out how to do that with a flow cell to ensure that it's 100%. I say 100%, to as close as you can to being 100% sure of the accuracy of your measurement.
All right. So Jay, we got a bunch more questions, a couple easy ones here, I think. So what strategies did you use, I guess, if any, to, I added that part, to reduce air bubbles from entering the VPE VPX?
Okay, yeah, that's something, I know people get obsessed with air bubbles. I think that's a question we, I don't think, so that VPE experiment we showed, actually it looked like there may have been some air bubbles trapped in the VPE, which is some of the cause for the readings being a little bit askew. But I don't know how, the way that these instruments are designed, especially the VPX, where the readings are is at the very bottom. So just by gravity, any air is going, it's at the very, very bottom of the flow stream. So it's kind of physically almost impossible for air to get trapped in say in the 10 micron bottom space of the flow stream, if the flow stream is say like a ½ inch tall or 10 millimeters in that case. So I haven't seen, I know with the VP I have seen issues where the air bubbles cause like variations in reading. With the VPX I haven't witnessed that yet, and like I said, I think it's something, as long as you bleed your system carefully, there shouldn't be any introduction of air into the system. It should be a closed, like fluidic system. I don't know if the VP has capacity to cavitate either. Too. That's something that if they know what cavitation means, you basically pull on a liquid and it creates like an air pocket. The VPX, because it uses, especially at high concentration, such a narrow physical range of the fibrate, I think that any potential for cavitation should be minimized. So in a well-bled system, I don't see air, and like I said, I don't anticipate air is going to cause any issues. And I've seen it occasionally with the VPE, but like I said, it's just mostly caused because of poor bleeding of the system.
Okay, so another question here. Will you use SoloVPE or A280 as offline backup assay? Or some other assay for an offline backup? Or maybe, I don't know.
Oh yeah, it's kind of a quality question. I'll say that there's been acceptance. SoloVPE is used for in-process, call it in-process release, and so it depends on the. It's one of those things like it's not a platform policy we have across like all drug substances. It depends on where it's manufactured, what the asset is, like basically how the process was developed. For some of the processes, so they're allowed to use Solo because they've done the qualification and demonstrated comparability. If that hasn't been done, then you're stuck with the A280. But I think there's right now the state of the art would be the Solo, but it's still an offline measurement. Ultimately, in a perfect world, the VPX could do it all online, but that requires some engineering to put it into basically the final. That's something we haven't overcome. We're actually thinking about ways that we can measure the recovered material without adding a second instrument, because I mentioned to Ramsey, I try not to say the limitations of the instrument, but one of the limitations of the VPX is the expense. Like each one of these units is they're not terribly expensive, but they're not cheap. And I think that if and the instruments are not, they're not big, but they're not small either. It requires a cart, that's, as you saw the size of the cart. So as the years go by, I think Repligen is working on, and we've been do some. If they create like a smaller detector, smaller footprint, like a smaller light source, that would be very helpful. So that potentially, if the price comes down and the footprint comes down, you can put these in a lot more space parts of the process. But right now, I think that the price and the size and the complexity, that's the reason why it's basically UFDF first, because that's where it's going to have the biggest bang for its buck.
Cool.
Answer the question. I went with straight.
Yeah, I think you did. Yeah, it's okay. Jay, go a little astray. Yeah, I mean, it sounds like the FlowVPE is a suitable backup in most cases. It depends on the molecule. So does extinction coefficient for a given mAb vary substantially between different formulations? And if so, do you adjust for that during your dye filtration?
Well, it should be relatively independent of the formulation. So that, I remember Ramsey, we early on we discussed this. I was thinking that extinction coefficient is concentration dependent, and then it starts getting a little bit weird because then if you look into the papers, how do you measure protein concentration accurately? I remember there was a group you said that it burned it, which was kind of like a little extreme. Trying to get the absolute protein concentration measurement. So to jump back to the question, I'm not aware that the significant variation in, I mean, maybe I'm not biophysically, I haven't studied it, but how much the extinction coefficient changes in different matrices. I think basically the way to get around that obviously is when you do your comparability testing. Your comparability testing is done with exact material. So that, like when I showed the data comparing the A280 to the flow and the solo, it was done in formulation buffer. So basically, and using the exact same extinction coefficient in all three methods. So based on at least a limited amount of experimental data and a limited amount of monoclonal antibodies, it appears that you're within 3% of the solo and the VP up to 220 grams per liter. And we haven't seen the need to basically to use a different extinction coefficient. The only thing I'll mention one thing. One of the beauties of this too is the VPX is that in order to get an accurate A280 measurement, you need to know the density accurately at every single concentration. In fact, that adds a source of error because it's almost like a chicken and egg or an iterative process. In order to know what the density of the material is, you need to know the concentration. But how do you know the concentration if you haven't measured the concentration? So because there's these charts that show how the density changes between concentration. So when they run the A280 test in the QC, they're using an approximate density to do the calculation, not the actual density because they don't know what the concentration is. So basically, you got some approximate. When you can measure it directly, the VPX, it just eliminates that whole issue of like having to know the density. Another thing too is the density changes with different matrices. That's the biggest issue, I think too, is that when you are dye filtering it, you're changing the actual density. So that would require that if you're doing an A280, you need to know the density in that matrix for the protein. That's to me the most significant factor or potential source of error when using an A280 method versus like extinction coefficient. Like as far as I know, I said should not vary too much. Some people say it changes at higher concentration due to like biophysical changes, but I don't think it's that significant.
Great. Yeah, thanks for that answer. So I have a comment here. Thanks, Jay. Very nice presentation. Great work with the FlowVPX development. The question is related to general absorbance limits for optimal data with high concentration material. Is there a target absorbance value that you tried to stay below with any of the variable path length technology that you evaluated?
Well, truth be told, the variable path lengths, this is the only, I don't know if there's any other in the market. And we've tested some other, like I know like the refractive index, and people know, I won't go into limitations of that, but as a general, so if anybody's familiar with it, there's an algorithm for these VPE instruments where it targets the middle set point path length. It runs the algorithm and tests what path length will give you an absorbance of 1, and then it chooses like a number of points, an equal number of points to spread on either side of the absorbance of 1. Based on our experience, above like 1.8 or 2 is when Beer's law becomes nonlinear. but up to it depends on the protein too. So it seems like some proteins become nonlinear at like early at a lower absorbance than others.
Yeah, so it sounds like that's something that you should probably evaluate on your particular material, right?
Yeah, and it becomes, I mean, you can go down a rabbit hole with like biophysical stuff, like molecular crowding and all that. But that's why I think it's, before we worry too much about that, if you can just directly test by A280, and like consistently see that like the VPX and the gold standards match, then I think you're okay. Because honestly, that's another thing too that I'll say that one of the reasons this maybe for the slow adoption of VPE is that a lot of people, there's a lot of I don't want to say unsubstantiated, but if you ask a lot of people in the past and even today, they'll tell you that the VPE is not accurate at high concentration, which I can see based on my experience that I don't see that to be true. There are limitations and you have to be careful though. Like I said, there's in a carefully controlled experiment, I see typically within 3%. So that was I think another issue that's been holding up like use of this technology, like in terms of being accepted, like because quality has to buy in, and if certain people have this perception that the technology doesn't work at high concentrations, then they're automatically sort of knee-jerk reaction is that, oh no, everybody knows that VP isn't accurate at high concentrations, whereas it seems like the evidence shows that that's not true. At least, especially this VPX.
Great. So that's all the questions we see here on the line. We're getting close to the end here, but if there's any more questions, please put them in real quick and I can relay the message. If not, thanks again, Jay. It's been really great. Again, I appreciate the work we've done with the VPX. I think it's been awesome. Next week we'll have Raf Dedier from Janssen presenting on his work with the drug product, and then the week after that, the panel discussion with Tony Hunt. So please join, please register, and if you want to watch the old ones, hop on the registration page, and you can watch the last two in this series. So again, thanks a lot, Jay, and thanks everyone for joining. And at this point, I guess we can close the call.
Yeah, thank you. Make sure you attend Raf's talk next week. I'm looking forward to that too. Thank you, everybody. I appreciate it. Thanks, Ramsey. Thanks, everyone.
Yep.
Bye.
Bye.